Hello viewers, dear students, my name is Bhatt Rafi. I am lecturer in zoology subject and my YouTube channel is Bhatt Rafi's Lectures. You can subscribe and share if you like the video. So today's my topic is lysosomes for class 11 students. So dear students, let us start the topic lysosomes. It is origin, structure, function. So let us begin this topic. You all know that lysosomes are also called suicidal bags. It is called suicidal bags or lytic bags or lytic bags. Lysosomes are the Greek word. It is the Greek word lysosis means digestive or loose, and somo means body. So it is a Greek word. Lysosis means digestive or loose. Somo means the body. The lysosome was first reported by Bill Gann. It was first reported by Bill Gann, cytologists. It was first reported by Bill Gann, cytologists and biochemists and biochemist Christian to do. And this lysosomes this organelle was observed under electron microscopes by scientists, namely Novakov in 1956. So lysosomes, if you go by the acarness of lysosomes, where is acar in the cell, where it is situated in the cell, you know that lysosomes, they are a simple, tiny, spherical, sac-like structure. They are present in cytoplasm of eukaryotic cells. They are present cytoplasm of eukaryotic cells. Eukaryotic cells. Each lysosome is a small vesicle surrounded by a single membrane and contains a powerful enzyme. It contains a powerful enzymes. These enzymes are capable of digesting or breaking down all organic materials, and lysosomes enzymes are made up of rough. Its enzymes are made up of rough endoplasmic reticulum. So we are talking the acarness. Lysosomes are practically in all cells, all animal cells and protozoans. It occurs in all animal cells and protozoan. And this a few type of cells such as mammalian RBC have a lack lysosomes. And mammalian RBC have not the lysosomes. Lysosomes have also been found in cytoplasm and certain plant cells such as yeast, fungi, root tip, cells of maize, cotton, pea seeds. However, they are less prevalent in plant cells. They are less prevalent in the plant cells than in animal cells. Prokaryotic cells are without lysosomes. I have already told you that prokaryotic cells have not possesses the lysosomes. It is present in the eukaryotic cells. It is prokaryotic cells are without lysosomes. If you go number of lysosome in a cell varies with the cell type. They are particularly evident in certain WBC macrophages, Kupffer cells, and secretory cells of pancreas, spleen, liver, kidney cells of degenerating or regressive tissues. For example, tadpole tail also contains many lysosomes. Tadpole tail contains many lysosomes. And this tadpole tail also contains many lysosomes. If you go structure of this lysosome, how it is made up of, you all know that structure of this lysosomes or small bag-like structure. It is small bag-like structure. Shape is often rounded. Shape of rhizosome is rounded or spherical. Shape is the rounded or the spherical. But may be irregular. If you go diameter, average is 4.4 μm to 8 μm. It is the diameter. In mammalian kidney cells, it may be as big as 5 μm, and is exceedingly large in phagocytes on the outside. It has membrane composed of lipoproteins, which is homologous to the unit membrane. In contrast to the cytoplasmic components, lysosome membrane is a single. The internal structure of lysosome is variable. Lysosome exhibits polymorphism on the basis of their content. There are 4 types of the lysosomes. First is the 4 types of lysosomes are recognized. First is the primary lysosomes. Second is the secondary lysosomes. And third type is the residual bodies or autophagic vacuoles. First we go for the primary lysosomes. Primary lysosomes, it's the first type of the lysosome. Primary lysosomes, newly formed lysosome. It is a newly formed lysosome, contains enzyme only. It is called the primary lysosomes. It contains its enzymes are probably in an active state. If you go for the secondary lysosome, if you go for secondary lysosomes, it is called the digestive vacuole. In a secondary lysosomes is called the digestive vacuole or heterophagosomes In a secondary lysosome is formed by fusion of food containing phagosome with the lysosome having heterolytic or digestive enzymes. Digestion occurs, the digestion of food passed into the cytoplasm. Finally, the secondary lysosome is left with undigested food. Third type is the residual bodies. Or we call it residual bodies. They are those lysosomes in which only undigestible food material have been left out. In an undigested food is removed out, thrown out. Undigested food have been left. The residual bodies are lysosomes and fuse with the plasma membrane to throw out the debris into the external environment by exocytosis. Process is the exocytosis. And third type is the and fourth type is the autophagic vacuole. Autophagic vacuole. A cell, autophagic vacuole, cell may digest its own organelles such as mitochondria, endoplasmic reticulum. This process is called autophagy or autolysis. I repeat is, the cell may digest its own organelles such as mitochondria, endoplasmic reticulum. This process is called autophagy or autolysis. Primary lysosomes fuse together about the damaged or unwanted organelles, forming a large sac known as autophagic vacuole or autophagosome. This is the autophagic vacuole. And if you go for origin of this lysosome arises from the Golgi complex. It arises from the Golgi complex. In a lysosome arises from the Golgi complex. Their membrane and hydrolytic enzyme are synthesized on the rough endoplasmic reticulum. And are transport vesicles to the Golgi complex for modification and packing. Secondary vesicles filled with the lysosomal bud off from the transface of the Golgi complex as the primary lysosomes. The protein of the inner surface of lysosomes membrane escape enzymatic breakdown, probably by having three-dimensional conformation that protect the vulnerable bonds from the enzymes attack. And now we go for the function of the lysosome. Function of lysosomes. Function is they help in waste disposal system and autolysis. They contain numerous enzymes which help in intracellular digestion or take part in intracellular digestion of foreign particles. And third point is they provide energy during starvation. In a lysosome, provide energy during starvation. And fourth is they help in defense against the bacteria and viral infections. And another is the lysosome removes cellular debris, or they help in removal of dead cells in tissues. And another function of lysosomes is lysosomes of WBC help in defense against cell infection by microbes. And they play important role in the metamorphosis of frog. This is another function. For example, disappearance of tail is due to lysosomal activity. And another function is lysosome cause the breakdown of aging and dead cells. So this was the lysosome structure and function. The rest is okay. Thank you all.